This one is nowhere apparently. Do you think it was just 3 hours? I think so. 8 lines, but rather than the company starts. Yeah. Let me tell you something. So funny for like this
Okay.
Something that people.
Hi, everybody. Here is exam 3 results, distributions. The raw score there, pretty wide range. The mean and the median were about 84. Standard deviation, I don't know. I think around 22 points, I think it said in the announcements. Let's see, what does it say in the announcements? Right, there we go. So mean is this, about 56%, median is 56%, standard deviation is 16.5%. Oh, this is the exam 3 advisory curve. Let me edit that. Edit. Just to prevent any. Okay. So I don't know, are the grades posted? They were going to be posted probably today, just so you get a sense. So it's about the same. It's basically a very similar distribution to exam 2. And, you know, if you have concerns, send me an email. Happy to give you some context as to where you stand overall in the course. Okay, so let's go to the, what else do we need to do here? Participants, let somebody else in. Okay. And let's go here. 28. Okay, so last time we were doing sexual reproduction and reproductive organs. I wanted to just sort of I've updated the slide a little bit. I wanted to go back to one aspect here, right? Which is related to the chromosomes and chromatids. Okay, so it's a little bit confusing, okay, sometimes, like what is a chromosome and what is a chromatid? So a chromosome, okay, it's just one long strand of DNA that's all coiled up on itself. It's packaged by histones, okay, and that's what's visualizable on that karyotype where you see the sort of plots of the 46, where they're like lined up 46 different sort of lines there. Okay, what is a chromatid? Chromatid is one half of a duplicated chromosome. So you can have one chromosome that has sort of two copies of itself, and you have the sister chromatids. Okay, that only happens at certain phases of the meiosis cycle here. Okay, so if we look back on like how many chromosomes do you have at each stage here? So if we let's go back to this one. Right. So in meiosis, right, we have a sort of duplication. So here is a case where we have diploid cell, but there's only one chromatid per chromosome here. Okay. Then we duplicate the chromosomes. We're still diploid, but now we have duplicated chromatids. Duplicated chromosomes, that means you have two identical chromatids attached to each chromosome. Okay, so then what's going to happen is those are going to line up. You'll get some crossing over. That's not shown here. Those are then going to split, okay, into a haploid with duplicated chromosomes. So now we have only 23 chromosomes, but they are duplicated. So there's two copies, okay, of sister chromatids on there. Okay, admit. And then finally, to make the final gametes, those are going to split in half, and we're going to have 23 chromosomes, 23 chromatids in each cell. So just to like make that a little bit clearer, I've laid it out and where that happens in each stage here in this cycle. So during this mitosis divisions, when you're in the spermatogonium, you have 46 chromosomes, 46 chromatids. So one chromatid per chromosome. Once those are going to duplicate, okay, have sister chromatids. So now we have 46 chromosomes, each with a copy of itself or a duplicated self. You'll have crossing over occur. Those chromosomes will then assort. So you get half of the chromosomes are going one way, half are going the other way, but they're still duplicated. Okay, and then to make the final germ cells or a final gametes, you're going to then split those chromatids or split those chromosomes into two chromatids. Okay, one into each of the sperm cells. Okay, then that's a question that came up during the Q&A session. All right, so let's go now to, where were we? So we, I think, stopped around here. So that same sort of diagram is going to come into play here, okay, on the ovarian development and oocyte development. The difference here is that when you have these divisions in meiosis, rather than having a symmetric division, one half of the genetic material is going to get thrown away into polar bodies, and each time it splits. Okay, and all of the other material, the mitochondria, all of the yolk, is going to split into, is going to be sort of asymmetrically distributed into one of the halves. Okay, and that's why we're going to be able to have a concentration of all of that supporting nutrients in the egg, which will eventually get fertilized. Another sort of thing to note, right? So the granulosa cells, you can think of them as the Sertoli cells. They're really derived from the same cellular types, right? They have similar functions. Thecal cells in females are equivalent to the Leydig cells in males. Okay, so let's now talk about the uterine or menstrual cycle. Okay, and this is something that happens around every 4 weeks, and it really is regulated by an interplay of 4 different hormones, really 5 hormones actually, if we're counting inhibin. Okay, but the levels of hormones that we're going to sort of see progress and change and fluctuate over the course of this 4-week cycle. So Since it's a cycle, you could define any time point as like the first day or the 0th day of the cycle. In this case, like the clearest external sign is going to be bleeding from the vagina. So we'll tell you which is associated with the deterioration of the uterine lining. So let's call that day 0, okay, down here, the first appearance of blood. So this is called the menstrual flow phase. Okay, so the first sort of period where a period is happening, you have blood coming out. That's going to be menstrual flow phase. Then there'll be a proliferative phase where the endometrial lining of the uterus starts to grow and regrow, developing lots of blood vessels, becoming thicker and sort of priming itself to be ready for implantation of a fertilized egg, if such a thing takes place. Then, sort of following ovulation, there's going to be a secretory phase where it's going to continue to grow, and then if it does not have the appropriate combination with uterine lining, doesn't have the appropriate combination of hormones, it will then degrade at the end of that. So what are those hormones, and what is all the steps here? So the first step is the release of gonadotropin-releasing hormone. Okay, this is coming from the hypothalamus. Gonadotropin-releasing hormone is going to promote the release of follicle-stimulating hormone and of luteinizing hormone. Okay, so as you can see here, right, GnRH coming from the hypothalamus hits the anterior pituitary. Those two things are going to be released. Okay, what is that going to do? That's going to cause follicles in the antrum area of the ovary to begin to grow. Okay, and they're going to start to compete with one another. Okay, and the granulosa cells within those developing follicles are going to increase their level of estradiol production and increase their level of inhibin production. So you can see that here. Okay, so these granulosa cells are producing inhibin to provide negative feedback on FSH, but they're also converting testosterone to estrogen, so they are creating a lot of estrogen. And the estrogen itself is going to start feeding back onto FSH, onto the anterior pituitary and hypothalamus to try to reduce the level of FSH being produced. Okay, so there's a sort of a bidirectional communication here between the anterior pituitary and hypothalamus to the follicles. So the follicles are producing hormones that are influencing the release of FSH from the anterior pituitary, and the anterior pituitary's FSH is influencing the development of the follicles in turn. Okay, so as the estradiol levels rise and the inhibin levels rise, this is going to provide negative feedback on the release of FSH by the anterior pituitary. Okay, now that is going to allow the follicles that are most sensitive to FSH to continue to grow, and it will prevent other follicles that are less sensitive to FSH from growing because the FSH levels are falling. Okay. Now there's a certain level of estradiol concentration where it reaches a level where estradiol is now, instead of inhibiting release of GnRH, it actually crosses a threshold where it starts to have a positive feedback on the release of GnRH, which then will start to spike the activity of these gonadotropes in the anterior pituitary and cause a surge in the release of luteinizing hormone. Okay, why does that happen? It's probably different receptors within the hypothalamus that have different affinities for estradiol, and they have distinct outcomes for whether or not when they get activated. Okay, so we have this sort of now, when we're at high levels of estradiol coming from the follicles, okay, we have this increase in FSH and in particular of LH, luteinizing hormone. You have this surge. That LH surge is what is going to push the follicle over the edge to eventually to finally rupture and release the egg. So that's what the trigger of ovulation is, okay, this LH surge. Now, after that LH surge takes place, we're going to have a drop in FSH levels. We'll have a drop in LH levels. Okay, And we're going to also have a drop in estrogen or estradiol levels because the granulosa cells have kind of been disrupted. Some of them are released from the ovary. And what will happen then if our estradiol levels fall? Okay, we now move back into a regime where estradiol is inhibitory to the hypothalamus rather than being stimulatory. Okay. Now, this is then going to, those levels of estradiol as well as the increase in progesterone is going to be what prevents a second ovulation. So where does that progesterone come from? That comes from the corpus luteum. Okay, what is the corpus luteum? It's what's left over from that dominant follicle post-ovulation. So it turns into those granulosa cells, and thecal cells sort of turn into this other sort of like mini organ, okay, that is going to be releasing, it is going to increase its release of progesterone, okay, and that is going to maintain the uterine lining and prepare it for implantation up until there's a failure of implantation to take place. Okay, if there is implantation, then it's going to maintain those levels higher. But in this case, in a sort of on your average cycle, in your typical cycle, right, you do not become pregnant, right? So so the progesterone levels, this is what's illustrated here. Progesterone levels go up from this corpus luteum, and then they are going to decay in the absence of some other hormonal signal, which will actually be coming from an implanted blastocyst, implanted fertilized egg. Okay. Okay. So estradiol and progesterone levels are rising. Estrogen is still suppressing FSH and LH. That's why we see this drop here in FSH and LH. Now, at some point, LH and FSH become so low that the corpus luteum, which is full of these cells that are responsive to FSH, starts to degrade. Okay, and so it will degenerate. That will then cause a drop in the level of progesterone and estradiol, and that will cause basically necrosis of the endometrium, which is this sort of rich lining of the uterus that's rich in blood cells. So when estrogen levels fall and progesterone levels fall, endometrium is going to deteriorate. Okay, so these endometrial cells, how do they deteriorate? They're going to release prostaglandins. Those prostaglandins, as you recall, often cause vasoconstriction. So that's going to basically choke off the blood supply to this thick lining, okay? And that's going to kill off, cause necrosis of the lining. Those cells will die, and that's going to lead to the menstrual flow phase, which is a sort of blood and dead tissue sort of flushing out, okay? Now, and why does that happen? It's really to maintain the health and optimal environment for implantation and refresh it every month. Okay. Now, what happens if implantation does occur, though? Okay. In that case, there's a different enzyme. There's a different hormone that's released by the embryo, the developing embryo, which is called human chorionic gonadotropin. Okay. And this is going to maintain the corpus luteum, which will then maintain the levels of progesterone, which will then sort of promote the maintenance of the uterine lining and cause like that lining to secrete glycogen that will provide nourishment to the developing fetus. Okay, so HCG, this human chorionic gonadotropin, that's derived from the fetus itself, okay, or the developing fetus. And that is the first sign of pregnancy. If you have very low levels of typically you have very low levels of HCG. Women have very low levels of HCG, so do men. Okay, as soon as implantation occurs, those levels spike up by 100 to 1,000-fold. Okay, which makes it the key signal that is used in pregnancy tests to identify at the earliest stage whether or not a pregnancy has occurred, okay, if conception and implantation has occurred. Okay, the way that we do that is by using immunoglobulins, conjugated to these sort of strips of material. If you urinate on that, then flow of that urine up, there will be some residue of that hormone within the urine. Flow of that comes up, and if it binds to these immunoglobulins for HCG embedded on the strip, it'll light up or it'll change color, and you'll see that pregnancy has occurred. Now, if a guy has peas on this strip, generally speaking, you won't see anything unless, and if you do, that's a pretty good sign that there's some sort of cancer, testicular cancer, that's causing an inappropriate generation of human chorionic gonadotropin. Okay, so this process of follicular development occurs every cycle, every 4 weeks or so. Okay, so how do we only get one egg released per cycle? Okay, so there's going to be a phase. Okay, at the start of the menstrual cycle, there's going to be a group of these antral follicles. Okay, those that have already formed a fluid-filled cavity. Okay, those are going to get recruited by the FSH levels. Okay, so all of those follicles express FSH receptors on their granulosa cells, and they are going to start to grow. Okay. Now, at the second sort of phase here, after about a week after the initiation of this cycle, okay, we are going to select a dominant follicle. That is going to be the one that has the highest sensitivity to FSH, probably because it has the most FSH receptors expressed on it, or has the most granulosa cells. It's already gone through the process of development. It's already sort of grown a little bit from prior cycles. So it's the most sensitive. It's going to be then producing, if it's the biggest, most sensitive, it's going to be producing the most estradiol, the most inhibin, okay? And that's going to feed back onto the pituitary and suppress the FSH levels. Now, as that continues to grow, the thecal layer, where all the thecal cells are going to become vascularized, which is going to increase the access of the developing follicle to the blood supply. Okay, so we need to have blood coming in and into that area. Okay, if it has better vascular access, it's going to be able to more easily pull the FSH that's available onto its receptors and activate itself. Okay, so that is basically going to allow it to outcompete the other follicles. Okay, it's going to be using more. It's going to be more efficient at using the FSH that's available. Okay, and it's going to start producing more and more estradiol and inhibin, which is going to lower the levels. Right? It's going to start dropping the levels of FSH, but it can continue to grow because it has the greatest access. Okay. Now, that drop in FSH levels is going to cause something called atresia, which is the sort of the reversal of the development of the other follicles. They are going to start to shrink back down, okay, because they don't have enough estradiol and autocrine, paracrine support to keep growing. Finally, once the dominant follicle becomes large enough and that high estrogen output of it is high enough to switch the mode in the hypothalamus to a positive feedback mode for estrogen, that's going to cause the surge in the release of GnRH, which then will cause a surge in the release of luteinizing hormone and FSH. The surge in LH is what's going to drive the final stimulation of the follicle to erupt and deposit its oocyte into the fallopian tube. Okay. I don't know why I forgot to write the sentence. I was changing this title. I forgot. I guess I forgot to. Here is the actual plots across the population, human population, of the 4 major players here in the hormones. Right? We've got estradiol. Okay, because you can sort of see from this chart, this is just some line that the people drew in a textbook. Right? Here's the actual data. Okay. And it's sort of hard to see some of the subtleties when you're just looking at that line. So for example, here, so we've got estradiol here, we've got luteinizing hormone, we have progesterone, and we have follicle stimulating hormone. So there's some cool things that you can see here. So estradiol levels really just continue to ramp up. Basically, estradiol levels are very sort of similar. They really follow the development of the follicle quite well, right? The more granulosa cells you've got, basically, that are in there, they're going to keep producing estradiol, and you're going to keep seeing that go up. Okay. One interesting thing here to see about that follicle stimulating hormone is it sort of initially peaks around 4 to 5 days in, and then it drops. And why does it drop there? It's dropping because a dominant follicle has been selected and is now releasing so much estrogen that it is sort of suppressing, it's releasing sufficient amounts of estrogen that is suppressing the release of FSH. Okay. Now, at some point, that follicle becomes quite strong, quite large enough so that the estradiol levels reach a critical threshold that then causes a positive feedback on the hypothalamus, and then we get the surge in FSH as well as the surge in LH. We call it both the FSH and LH go up there, but we call it the LH surge because LH is really like it's really flat up until a day or two before the ovulation event occurs. So that's really the key sort of differentiator there. Okay. What else can we see here? The progesterone levels are basically zero until you get the corpus luteum. The corpus luteum is what's producing the progesterone. The progesterone is what's going to tell the developing uterus to really start secreting glycogen and things like that are going to really support any embedded embryo within it. Okay. And in the absence of the corpus luteum, when the corpus luteum begins to degrade, we see that drop here. So, and one interesting thing also about this is the error bars here, right? There's a lot of variation, even from cycle to cycle, right? So, sort of these inner sort of bars here are cycle to cycle, and then across different women is here in the outer error bars, and we're zoomed in here, but there's massive amounts of variability from cycle to cycle. This can lead to, variability in the ability to conceive on any given cycle, as well as timing of cycle, and many different factors and hormonal factors can influence this. Overall levels of body fat, overall levels of stress or exercise can cause failure to ovulate on a particular cycle. So, but here's the real data. Okay, any questions on these cycle stuff? Okay, cool. So let's now look at the sort of structures here that are associated with conception, right? So we've got the fallopian tube and the ovary. So ovary is here. The ovulation is going to occur into the fimbriae of this uterine tube, okay, also known as the ovarian tube. So here we go. So the ovum, so this is a single egg, is called an ovum, is going to pass into this fallopian tube, into this like basically funnel, okay? And that funnel is called the infundibulum, and they've got these little fingers on it called fimbriae. Okay, and those fingers are going to sort of move, and cilia along the wall of these fallopian tubes are also going to move and sweep the egg down into the tube. And so that's so those cilia are sort of on these epithelial cells that line the tube. And the fertilization event, if it occurs, is really going to occur up here in this top third of the tube. Okay. So that will allow development of the first few cell divisions to take place as the cell travels down here. And then if it's fertilized and it's started to divide, it will tend to bury itself within the endometrium, which is this inner lining of the uterus. Okay. And eventually be supported there. So we have, we've got our uterus. This is a sort of thick muscle that can expand dramatically during the course of fetal development, and also can contract during labor in order to basically squeeze out the baby past the cervix through the vaginal and out into the world. Okay, so it takes almost a week for the oocyte to reach the uterus. Okay, so if it does not get fertilized in that time, okay, actually it has to get fertilized pretty early on. It will start to degrade on its journey down here. Okay, so during the course of that journey, this endometrial lining will continue to thicken, okay, in preparation for an arriving zygote. But it doesn't really know that fertilization has or has not occurred until implantation takes place, because that is when the human chorionic gonadotropin will start to, levels from the fetus will skyrocket and then signal to the rest of the body of the mother that we have had a fertilization event and we have a developing embryo within our body. Okay. So if that doesn't occur, if we don't have HCG, we're going to have a degradation of the corpus luteum, drop in progesterone levels, drop in estrogen levels, and a sort of a degradation of the lining and menstrual flow. Okay. This is basically saying the same thing. just showing you where those sperm are meeting the egg. Okay. So you've got about, I don't know, 500 million sperm in an average ejaculate is deposited into the vagina. Only about 50 to 100 of them will make it to the egg. Okay, they get up there relatively quickly, although they can stay around and keep swimming for a few days, so they can still sort of, if a ovulation event doesn't occur immediately or hasn't already occurred, if it occurs shortly after the sperm entry into the uterus within another day or two, you still can have a fertilization take place. 50 to 100 sperm will make it up to that area relatively quickly, but only one is going to fertilize the egg. We have to have only one fertilize the egg because, you know, a sperm and an egg, they both have a haploid, you know, a haploid. They both have 23 chromosomes, 23 chromatids. We need 46 in order to make a human. We don't want more than 46. We don't want to have 2 sperm get in there, okay? Because we'll have an inappropriate number of chromosomes, okay? So how can we do this? Okay. There has to be a way first for a single sperm to get in, okay? And there's a series of reactions that are going to take place to make sure that only one sperm makes it, okay? So the sperm actually, there's a whole cohort of them, you know, 50 to 100 of them. What are they doing? They are initially trying to burrow into that layer of granulosa cells that are still sort of attached to the ovum, okay? So those follicular cells are surrounding the egg. They need to sort of get digested away, and they need to be penetrated by the sperm. And multiple sperm are going to be sort of trying to do that at the same time. They're all sort of competing to get in there once they reach there. And they're sort of working, their digestive enzymes are sort of working together to get past that initial barrier. Then we have this jelly coating, okay? When the head of the sperm here reaches the jelly coating, it can react with that jelly coating and release the acrosome, okay? So by that, there's going to be a sort of a reaction here between the jelly coat and there that will then release a new set of enzymes that will further specifically digest this particular jelly coat, okay? And then we have a receptor layer, sort of a bunch of receptor proteins here that are going to be able to bind specifically to the nucleus of the cell, of the sperm, and allow the passageway of the nucleus into the egg, okay? So we've got to get past the plasma membrane here, though, in order to do that. So there has to be some sort of fusion event that takes place. That fusion event is going to be dependent on the electrical potential of the inside versus the outside of the cell. This egg has to have a negative electrical potential. So the inside of the egg has to be more negatively charged than the outside of the egg for that fusion event to take place. Okay. Okay, where are we? So this proteins on the sperm. Okay, so the selectivity for species-specific fertilization, only, as I mentioned last time, only members of the same species can fertilize the egg, and that takes place because of the cohort of receptors along this plasma membrane layer. Yes?
The uterus is very big, how does the sperm actually find the egg?
So the uterus is very big. How does the sperm find the egg? Well, you've got 500 million of them there. So they're going to basically flood the zone. I don't know, you know, they're going to try to seek out every direction and swim in every possible direction. And a very small percentage of them, you know, well less than 1% of them is actually going to make it up the fallopian tube. Okay, so we've got this fusion now of the plasma membranes, and the sperm and the egg are going to fuse. That sperm is going to get delivered, the nucleus gets delivered into the cytoplasm of the egg, okay? And then that is going to cause two things to happen, okay? It's going to cause a fast blocking. This is the moment, okay? The instant the sperm nucleus gets into the cytosol through fusion with the plasma membrane, you have 2 reactions that are going to take place. One is a very fast event that lasts a short period of time, that instantly blocks any other fusions from taking place. Okay, this is called the rapid depolarization phase. Okay, so what does that mean? Rapid depolarization. Polarization means there's two things that are very different from one another, right? Okay, so a polarized cell has an inside that's very negative compared to the outside of the cell. The electrical charge on the inside of the cell is much more negative than the outside or of the membrane. So what happens is, the first step, there's a bunch of sodium channels, okay, that are on this outside edge of the plasma membrane. Those sodium channels all open up. Okay? The opening up of sodium channels will do what? It's going to allow sodium ions to flow into the egg. Okay? Sodium ions are positively charged. That's going to now make the membrane go from very negative on the inside of the cell to pretty much flat. Okay? Pretty much zero. Okay? It's going to depolarize the cell. That will prevent, that electrical depolarization prevents the fusion of any other nuclei with the plasma membrane, because the molecular species that allow that fusion to take place, that have that sort of configurational energy to do that binding and do that fusion event, require the inside of the cell to be negative. They utilize that electrical potential in order to give it the energy to fuse. So that is going to be the first step. That happens, depolarization, it happens like in milliseconds. Okay. However, it doesn't last forever. Okay. So you have to have a second reaction that is going to further allow a long-term prevention of further fusion of sperm nuclei with the egg. And that is called the cortical reaction. Okay. What happens there is we have second messengers, IP3. Okay. An called triphosphate is going to be formed within the plasma membrane there, and it's going to cause the release of calcium from the endoplasmic reticulum. Okay. That calcium is then going to cause a release of vesicles along the cortex, the outer area of this of the egg, to then fuse here and release enzymes that are going to harden this jelly coat and is going to prevent any other sperm from being able to burrow in. Secondarily, okay, this vitelline layer, so this sort of layer here, is going to lift away from the cytosol and the plasma membrane of the cytosol. And that's going to provide a gap between the cytosol and the outer layer of the coat, just in case a sperm does somehow get through this jelly coat, it still will not be able to eventually reach the cytoplasm of the egg. Okay, so this this cortical reaction event is congruous at the same time, triggers the second meiotic division, and now we're going to have basically the egg cell is going to spit out half of its remaining chromosomes into the second polar body, okay, into a tiny little like bag that's going to then get degraded, okay, and the rest of it, we have a haploid 23 chromatids that are then going to fuse with the nucleus of the sperm here and to form this zygotic nucleus. Kind of complicated. Any questions about this? Yeah.
What's the cortical reaction? Does it happen? Like, I mean, the nucleus of the sperm integers, or
it happens at the moment of fusion of this nucleus, sperm nucleus, with the plasma membrane of the cytoplasm is what causes the electrical depolarization. Okay, that happens in milliseconds. That electrical depolarization triggers the production of IP3. That's going to take on the order of seconds to then cause the release of calcium from the ER. So it's going to happen within a few seconds of fusion of this sperm nucleus with the cytosol. Okay. Okay, so then what happens after that? Okay, so fertilization takes place. A miracle has occurred, right? So what then happens? Well, let's follow the path of development in the first few days after fertilization. And they're basically the terminology for all of these sort of things change. So at the beginning, now the egg is really big, right? It's got a lot of yolk. Why is it big? It's really so that, it can have enough space for cellular division to take place without any growth of the overall egg itself. So, in these initial events of development are all going to be called cleavages. So the cell's just going to divide in half, okay? And then those halves are going to divide in half again. Those halves will divide in half again, and those halves will divide in half again. So we're going to go from one egg, okay, one cell, that's divided 4 times into 16 cells, but all within the same sort of structure there. So fertilization is happening in this fallopian tube within a day or two within about a day of ovulation. The first cleavage will take place, and the second cleavage will take place as it travels down the fallopian tube. In this case, in those first few divisions, every cell looks like every sort of cell within that zygote, okay, looks like itself. However, once it reaches this morula phase, you start to see some differentiation of the shapes of the cells and the positioning of the cells within the sort of developing, I guess what we call it, an egg, within the developing morula, let's call it that. So here's what the zygote looks like. So following fertilization is called the zygote. We have a cleavage of the zygote. This is the 2-cell stage. We've got our sort of big cells here. We have our first and second polar bodies here. These are going to get degraded, okay? And these are going to be sort of the duplicated genomes of the first split. Then we have the 4-cell stage at day 2. The morula stage is now this sort of solid mass of cells, and it's then going to turn into a blastocyst, which causes a fluid is going to fill up on the inside, and it's going to basically turn into like a shell, okay, of cells. And this is going to allow a sort of an asymmetric differentiation here of cells within the inner mass and this outer ring of cells. So the inner mass, this is what's going to become the baby, right? This is going to become an embryo, and this outer sort of ring here is what's going to interface with the uterine lining and allow the implantation and burrowing in of this embryo into that lining. And this is called a trophoblast, and this is also what's going to turn into the placenta. So the placenta is actually derived, which is actually going to be derived from the sort of developing cells that are developing to turn into the embryo. Or I guess it's derived from the blastocyst. And so the originating point of the placenta is from the egg itself. Okay, so let's look at that implantation process. So here's our trophoblast. Here's our blastocyst as a cavity. We've got our inner cell mass. The trophoblast surrounds completely this inner mass of cells. Okay, so the trophoblast cells that touch the endometrial lining, okay, are going to sort of attach to it, okay, and then that'll cause some paracrine signaling here, and it's going to cause the trophoblast to start releasing digestive enzymes, which will allow the penetration of this blastocyst into the endometrial lining. So the inner cell layer is going to start dividing to form an embryonic disc, okay? It's going to cause, this is the first formation of the amniotic cavity, okay, which is going to be a bag of fluid that surrounds and protects the developing embryo and the developing fetus. And some of these trophoblast cells that we've got some of them are dissolving the material. Some of them are going to start providing this chorion. This is when the human chorionic gonadotropin starts to be released, okay? And that chorion is going to be what is going to eventually form into the placenta. So eventually we burrow all the way in, and this is sort of, the chorion is going to developing into the placenta. We have the amnion, which will become the amniotic cavity. We have a yolk sac here still. This is still providing nutrients. It's still not getting a lot of nutrients from the mother itself because it has not yet developed into a placenta, okay? But it's sort of approaching what are called lacuna here, which are going to eventually form along with the core, and they're going to sort of collaborate together to provide a ability for the capillary networks of the mother to link up with the capillary networks of the baby, not directly, but next to one another, so that they can pass gases, nutrients, and even IgG immunoglobulins from the mother into the baby's blood. So the circulation of maternal and fetal blood is not in direct circulation, but there are these, within the placenta, there are areas of close apposition between those capillary beds. So material can be passed from mother to baby. And in fact, hormones from the baby get passed back to the mother, and that's what actually controls the timing of labor and delivery. Okay, so what are some things that can go wrong? So endometriosis is very common. The endometriosis is where the endometrial lining, okay, which, has to have the endometrial cells, they have to be able to continue to do mitosis and proliferate throughout basically the course of a woman's life, at least for at least a portion of the fertile portion of a woman's life. So you can imagine, though, if those cells migrate to an area which is inappropriate, then the hormonal fluctuations that cause the division of these cells can cause the growth of that material in areas that no longer shed it, right? So if the endometrium migrates to places like the fallopian tube or the ovary, okay, or other parts of the abdomen, okay, you'll have these growths and fluctuations in size that are not shed later on, okay? So that can cause, it could be asymptomatic, or it could cause very painful cramps, irregular bleeding. Then we have, so that's endometriosis. Another thing that can happen is an ectopic pregnancy. So if you have an implant, if the egg gets fertilized, okay, but then it implants in the wrong spot, if it implants early, right? Not everything's perfect. Maybe it gets stuck here in the fallopian tube, and the trophoblast starts trying to burrow into the fallopian tube. This is not a good situation, okay? So this is going to result either in a miscarriage or a surgical intervention to prevent it. It can be a dangerous situation because the developing baby cannot, or this developing, not baby, but embryo is going to cause a sort of physical strain on the fallopian tube, and will cause it to rupture unless you have medical assistance. And then finally, we have hysterectomy. So this is the removal of the uterus and ovaries. Sometimes this will occur, if there's uterine cancer is detected, or if there's very painful endometriosis as sort of a medical intervention there that will sort of block that pain and prevent the irregular bleeding by removal of the uterus and the ovaries. That's also, of course, that's going to impact hormonal cycles. Okay, so that's basically how it works normally. Now, as a treatment for infertility, there's in vitro fertilization was developed by Robert Edwards and Patrick Steptoe. So in this case, they take the eggs out. Basically, you harvest eggs through a process of sort of hormonal control. Follicle development is hormonally controlled. So if you do a particular pattern of artificial hormone delivery, you can cause development of the follicle and of multiple follicles. Those can be harvested, okay, extracted, and then mixed in a test tube with sperm, okay, from the father. Now, after if we get fertilization to take place, you will get hopefully more than one, usually, maybe a dozen or more blastocysts that are now in this test tube. And at that stage, after a few cell divisions, they're taken out and frozen, okay, or to be saved, or some of them will be implanted into the uterus of the mother. Okay, so now when they implant them, they don't implant one usually. They usually implant, I don't know, 10. It's because the odds that any one actually implants are relatively low, and the odds that one will come to term are relatively low. So because of that, and because you're sort of really hoping to get at least one, oftentimes you'll get two or more that implant. Okay, so in the case of twins, like the natural incidence of twins is very low, relatively low, about 1% in sort of natural occurrence. But the fraternal twins, that is twins that come from two different eggs, okay, is actually relatively high in vitro fertilization just because of how the sort of how they're trying to the process tries to maximize the odds of at least one child developing. Okay. Any questions on that? Yeah.
What causes it?
I mean, the cells are under endometrial cells are constantly sort of every month they're under this great fluctuation in hormonal control, and over time cells can migrate and proliferate in different locations. So once they've proliferated into an area that doesn't shed, they're going to stay there and continue to grow and shrink over the course of many cycles. So it can progress. Okay, so here's so we've embedded now into the uterine wall. The baby's developing. We have a placenta is now formed. By week 8, we've basically looked like a little baby human. Okay, even though we're still quite small. So this is 5 weeks in, right here. You can see a little tail, lower limb bud. These other limb buds, an area where the heart is going to develop, an area where the eye will develop. Things that used to be gills. Okay, that's out like 5 weeks, but by 8 weeks, we start to really look like a baby. Okay, this tail has disappeared. The feet and arms now look like real limbs. Eyes and nose and ears are all starting to develop. Okay, so at that time, also the gonads have been formed, and all the major organs are present, but they aren't really working yet. Okay, now it's kind of interesting, this sort of transition between the very early embryonic development and then sort of mid-embryonic development. And a term that you probably you'll hear many times throughout biology is ontogeny recapitulates phylogeny. 3 really big words, right? What does it actually mean? It means the development, now, when you hear this, you can know, oh yeah, of course, ontogeny recapitulates phylogeny. What it means is development during the development of the embryo is actually going to reflect the the evolutionary history of the entire species. So all basically vertebrates look very similar at the earliest stages of embryonic development. Okay, we all have got gill slits, developing gill slits. We all have a tail, but during the process of development, okay, there's going to be a differentiation that takes place in mammals and humans that gets rid of those things. Okay, that suppresses them, and that's probably because of sort of the evolutionary history. Some of those genes to produce those things are still there. The overall sort of recipe for development is never like rewritten completely, right? We're just making edits to it over time. So maybe we have a new gene that's develops that suppresses or causes the regression of a component that's no longer needed for our overall growth to a human. Okay. Okay, so let's turn now to any questions on this. Okay, so let's now look at the development of sex organs, and how hormones and genetics influence the development of the sex organs. So early on in embryonic development, doesn't matter if you have an XY gene or an XX gene for your sex chromosome, or XXY, okay, which would be unusual, but does occur, okay. The gonads will be bipotential. That means they can turn into either testes or ovaries, okay. Additionally, there will be 2 sets of ducts that come off of those bipotential gonads. There's a duct that is associated with male sexual organs, which is the Wolffian ducts, and female organs, which are called the Mullerian ducts. Okay? And then the sex hormones that are produced are going to regulate the formation of these internal structures and promote the development of the gonad into testes or ovaries and allow the persistence of one set of ducts and the degradation of the other set of ducts. Okay, so in the case that you have an XY chromosome, so you will always get an X chromosome from your mother because your mother can only give you X or X, right? You're going to have a 50% chance of X or 50% chance of the other chromosome getting sorted into you. So it's X or X. You're always going to have an X from the mother. But from the father, you can have a 50% chance of getting the X chromosome, in which case you'll become female, 50% chance that you'll get the Y chromosome, so you will develop into a male. Now, of course, so it's really the father's genetics that determine the sex of the child. This was, news to Henry VIII because, he went through like 6 wives trying to finally find one that would give him a male heir, but never did it, even though because it was all his fault. He was only giving the X chromosomes out. So if you have XY, the XY has a SHY gene, which is going to cause the differentiation here into the testes. So the testes, once we have a differentiation and development of the testes, those are going to start to make testosterone. Okay, and the testes will also make this anti-Mullerian hormone. Okay, that is then going to cause the degradation of the Mullerian ducts, while the male ducts, the Wolffian ducts, will be maintained. Okay, so this sort of process here, testosterone and AMH, are going to lead to the maintenance of the Wolffian ducts, the differentiation of those ducts into the vas deferens, the development of the seminal vesicles, the prostate gland, bulbourethral gland. All of those things are going to be determined by testosterone. Then, in the absence of male hormones, if you never turn on the SHY gene, if you never then generate large amounts of testosterone, then these bipotential gonads will differentiate into ovaries. The Mullerian ducts will differentiate into the oviduct, the fallopian tubes, and we'll see the development of the vagina and uterus. Okay. So, which are connected here to these female ducts. So 2 different paths, XX or XY. How do we actually determine how these hormones generate these 2 different pathways? Cause these 2 different pathways? Well, scientist Alfred Jost, they took bunnies and basically castrated rabbit fetuses at different stages of development to see, what were the actual the sources of the signals that caused differentiation into male or female based characteristics. Okay, so in the first case, we have this undifferentiated case, right? This is our undifferentiated gonad early on. Okay, it has bipotential. It can become testes or it can become ovaries. You have the male Wolffian, the female Mullerian ducts. They're still sitting. They're both there early in development. They're present in parallel. Okay. Now, if you have an XY chromosome, okay, sort of our control male, what will happen is the Wolffian duct develops in the vas deferens, seminal vesicles, ejaculatory duct. The Mullerian ducts degenerate. If you have XY chromosomes, the opposite happens. Mullerian ducts are going to develop into fallopian tubes, uterus, cervix, vagina. The Wolffian ducts will degenerate. Okay. So what happens if you castrate an XY, castrate the male, okay? That is going to remove it's going to deprive that embryo of AMH, anti-Mullerian hormone, and testosterone. Okay, if we don't have AMH, that is going to allow the Mullerian ducts to stay around, the blue ones to stay around. Okay, and the Wolffian ducts will degenerate if you don't have testosterone around. So this male fetus is going to develop female internal and external genitalia. On the other hand, if you have XX and you castrate, you're going to have a very similar situation. Okay, so you're going to still have Mullerian development. The Wolffian ducts will degenerate because there's no testosterone, basically, this basically shows that the ovary is not required for female duct development. Yeah. It's basically the default pathway. Now, if we take a male, XY, and we castrate only one side, okay, so remove one testes, what's going to happen there? That's going to be kind of weird. Okay, on that case, the side in which the testes or in which the gonad is spared actually allows the maintenance of the Wolffian duct, okay, and the degradation of the Mullerian duct on just that one side. So that sort of tells us that there's some local signaling going on here. It's not just something that a hormone is circulating throughout the body, right? It's actually, there's probably some paracrine signaling that's required here because we have this differentiation between one side and the other. Okay. Now, what happens if you have an XY male? You castrate, but you give them testosterone, okay? Because the testes produces testosterone. And this is how we're going to see that the impact of testosterone is distinct from the impact of anti-Mullerian hormone. Okay, so the testes make both. Okay, if you if you remove the testes but apply testosterone sort of universally, that will preserve the development of the Wolffian ducts, the male ducts here. Okay, but because we don't have any AMH from the testes, we got rid of that. The Mullerian ducts stay around as well; they don't regress. Okay, so now we have two sets of ducts. I think it's going to be very confused what happens, probably in the genitalia there, but that is the case of testosterone during early development when you don't have any gonads around. Okay, finally, if we have female XX genes treat with testosterone early on, what happens? You have the testes develop into ovaries. The Mullerian ducts are maintained because there's no AMH, because these are not testes, they've developed into ovaries. But the testosterone presence allows the Wolffian ducts to also be maintained. Okay, so this is how they sort of broke down the differentiation between the role of testosterone, which is supporting the Wolffian ducts, and the role of anti-Mullerian hormone, which degrades the Mullerian ducts and then causes this sex segregation of genitalia. Yeah. Right. So it really is going to depend on, what sort of intersex chromosome you'll have. Sometimes you'll have XXX, sometimes you'll have XXY. And that can lead to different levels of testosterone levels, different testosterone levels. There's a variety of different outcomes there, and can lead to sort of, genitalia that might look sort of in between. And this is the, basis of, a lot of, transsex development, right? So it all comes down to, what are the hormones? What are the sort of genes that you have, or XX or XY, or a different cohort of those, and how does that intersect with the hormones that are developed by the gonads during that course of development? And of course, during puberty, it's a whole nother level of hormonal development and generation of secondary sex characteristics that take place. So, hormone therapies pre-puberty have a dramatically different impact on the development of a person than post-puberty. Okay, so let's see here. So now we have labor. Okay, how do we get labor? How does the mother know that it's time to deliver a baby? Okay, now the thing is, a baby is developing baby is very relatively taxing on a mother, right? It is taking up a tremendous amount of resources, okay? It is trying to get as much glucose as possible to develop from the mother. So it sort of promotes high blood glucose levels. It's going to try to suppress insulin and raise blood glucose levels in the mother. This is why you're often going to see gestational diabetes take place during development. That's because of signals coming from the baby, hormonal release from the baby in order to try to raise increase the level of blood sugar within the system. And over time, okay, the baby is going to be the source of hormones that are going to initiate the sort of early phases or pre-labor conditions. Okay, so how does that happen? So we have ACTH that is in the fetal adrenal glands are going to secrete hormones, cortisol and DHEA, okay, that are going to come over here, enter the placenta, cross over into the maternal bloodstream, okay, and then impact the sort of development of hormones within the mother. So DHEA is going to stimulate the placenta to produce estrogen. Estrogen is going to increase the amount of oxytocin receptors in the uterus, and that is then going to allow the uterus to become more sensitive to oxytocin and then drive the contraction of the uterus. Furthermore, the cortisol is going to stimulate trophoblasts in the placenta to produce CRH, which is going to stimulate the ACTH in the baby's pituitary, which is then going to sort of feedback and cause a positive feedback loop in this process. Okay, so we're going to get more expression of oxytocin, more generation of estrogen, more expression of oxytocin receptors in the uterus, and making that more and more sensitive to the presence of oxytocin. So as we sort of seen before, once the baby is in a position where it's putting pressure on the cervix, that's going to cause mechanoreceptors in the cervix to send electrical pulses to the hypothalamus. That will then release oxytocin from the posterior pituitary, which then travel and hit those oxytocin receptors on the uterine wall and start generating contractions, which then cause more force on the cervix, cause a dilation of the cervix, relaxation of the cervix, degradation or dissolving of a mucus plug that sort of blocks the cervix during the course of fetal development. And eventually dilation occurs up to about 10 centimeters. At 10 centimeters, that is where labor is going to actually take place. And this is when the doctor will tell mother to squeeze, start actually trying to push out and get active voluntary sort of a process in expelling the baby. Okay. Of course, oxytocin is also deeply involved in the production and in the letdown of milk. So the mother is going to produce milk in the mammary gland shortly after birth. Immediately after birth, there's already stuff in there called colostrum. Okay, this is sort of a nutrient-rich material, but that has basically immunoglobulins and other sort of things that a baby needs in a very short term to load it up. And that colostrum lasts for a few days. But it's really then replaced by milk, okay, which is a more, voluminous and nutritive substance. We have basically the impact. Basically, milk is going to be produced by prolactin release from the anterior pituitary. That is then going to cause production of milk, formation of milk, then suckling signals from the mother will then cause feedback into the hypothalamus, cause the release of oxytocin from the posterior pituitary, which then is going to cause the relaxation of certain ducts within the breast that's going to allow milk to come out. Okay, there's a positive feedback loop here between mechanical stimulation and release that's then going to be terminated when the baby gets full. Okay. Okay, so what are things that can go wrong with delivery? So if there's no sign of labor after 42 weeks, if the baby is getting too big, people are worried, doctor gets to maybe, and that's of course a stress on the mother, leading to more elevated blood sugar, could cause complications in the mother, also could make it difficult for the baby to squeeze its head past the pelvic bones of that surround the sort of surround the vagina, then you can induce labor via delivery of synthetic oxytocin, that's called Pitocin. So this is how the sort of timed pregnancies work. And so that's a very common, both in natural deliveries. If you want to progress labor, if it's sort of stalled at a particular point, they can apply Pitocin. Or if you want to induce labor to begin with and start that positive feedback process, you can deliver synthetic oxytocin to the mother's blood. Then we also have another complication is a breech baby. So normally, about 90% of the time during the course of development, during the last 4 weeks of development, baby's going to turn and face upside down to get the head into position so it can be expelled head first. And it's much more easy to sort of rotate the body and allow the baby to come out that way. The alternative is a breech birth where the baby never turns. Okay, this is going to be problematic. If the baby hasn't turned yet, and I can identify this through ultrasound. There's all sorts of sort of manipulations that a doctor might try to do in order to induce the baby to turn. Sometimes it doesn't happen. Almost always these days, because of that potential complications associated with that, much more difficult to get the baby out in this sort of breech position that will lead to a cesarean delivery. Okay, so what is a cesarean delivery? It's where the maternal skin and uterine lining is cut and under anesthesia, and the baby is pulled out directly, okay, and then sewn up. It takes longer to heal from that, but about half of the deliveries these days in America are cesarean versus natural delivery. Okay, so that sort of prevents the certain surges of hormones that are associated with the final delivery of the baby's crown out during natural birth, but it also is prevents some potential medical complications. Julius Caesar was not born by cesarean section, by the way. Couldn't have been, because his mother lived to a ripe old age, and very unlikely that you're going to survive a cesarean section in Roman times. Probably get sepsis and die from that. So really, we think that the origination of that phrase is from a Roman law, lex caesaris, which required babies to get cut out of the womb if the mother died during childbirth. Yeah. So they're only using it if the mommy's already dead. Supposedly. I don't really know what happened 2,000 years ago, but that's what they say. Okay, and then finally, okay, what happens after birth and in child development? Well, babies are going to grow very rapidly. The first week, you're going to see a drop, okay, in the weight of a baby, and basically you're going to the doctors are going to be checking the baby basically every day. Okay, weigh the baby, make sure. In the first few days, though, generally there's a drop because the amount of nutrition that a baby can get in those first few days from milk is not generally sufficient to maintain body weight, because the stomach of a baby is very, very small when it first is born. A newborn infant, day one, stomach is not big enough to drink enough milk to replace all their caloric needs. Okay, but within a week, milk production ramps up in the breast. The stomach's already expanded quite a bit. And at that point, the baby should be able to put on calories or put on weight from the milk. If it's not putting on weight from breast milk alone, then doctors will often try to feed it formula or have the mother feed it formula as a supplement. Breast milk is preferred because it has antibodies and all these other sort of hormones within it. But particularly as a supplement, formula is quite effective at causing weight gain. And then, you get to track your kids throughout sort of their life now and figure out where they live on these growth charts, right? So there's a rapid development phase in the first sort of 3 months. That's the time of your greatest increase in both the length and the weight of a baby, just insane, massive growth, which then sort of tails off into a steady rate of growth up until puberty. At puberty, there's going to be another sudden spike in growth rates, differentiation in secondary sex characteristics. But basically, you can predict this sort of in puberty, like the timing of puberty is very different from person to person, right? It can range across, 3 to 5 years. And so there's a great variability across the population in sizes around that time of puberty based on when puberty actually is taking place for each person. So the best predictor of actual height in a fully grown human is looking at their height at age 2 and just doubling it. Okay, because there's also great variability in sort of size of a baby at time of delivery based on how long they've been developing within the fetus and whether or not they're a twin. If you're a twin, you're going to be competing for nutrients from the mother, right? And when you are delivered, right? So, an average baby is maybe 7.5 pounds or 8 pounds at full term, but a premature baby could be, is now viable down to maybe even 2 or even less than 2 pounds. With all the medical technology that's been developed, the surfactants and things can allow those babies to survive. And eventually, they often catch up, but it can take them many years to do so, to catch up on those growth curves. So 2 years is the most predictive time for your overall height. And any questions? Okay. All right. Next week, we're going to do nervous system, the brain, motor systems for the last 4 lectures, which is, of course, like, my topic of research and study. So it should be good. Thank you all. What's up?
How are you doing?
I'm okay.
Just the last four.
Yeah, the exact final's not cumulative.
Okay. And then question about like the curve I did. Do you determine the stop the curve?
We all meet together.